All right, welcome everyone. My name is Pier Andrée, and I'm here to talk to you about some technical considerations in fluorescence-based cell sorting. You may have attended Fan Zhang's talk. She kind of did an intro to flow cytometry. So I will focus more on specific cell sorting technology. So my first slide, I hope, will show you a little bit about kind of the development of the fluorescence-based cell sorter over time. It's kind of over the last, now reaching 70 years in development, using various technologies, starting with the Coulter counter, an inkjet printer, and of course, merging that with some lasers. I will start with a little bit about talking about what is flow cytometry, and then I will move on to a little bit about the history of the development of the cell sorter. So flow cytometry is it's a sophisticated cell imaging technology, but unlike typical imaging technologies, it just looks at populations of cells that are hydrodynamically focused into a single cell stream, and it's pulsed with lasers to record fluorescence at different wavelengths. We don't actually get an image, any spatial information, but we do get mean fluorescence intensity. And then we use fluorescent dyes and probes, allowing us to analyze the expression of a variety of different cell markers. We can also look at organelles or DNA cell cycle, for example. And fluorescence-based cell sorting uses electrostatic deflection to purify populations of interest based on their fluorescent properties. We can also do it without any fluorescent properties. It's a little bit more challenging, but we can certainly try. And finally, cell sorting can generate purities of over 99%, making it still the most effective technology for fractionating out mixed populations. So just to talk a little bit about the outline for today, I'm going to start with the history. I'm going to then talk about some applications and measurable parameters. How does it work? And then a little bit of tips and guidelines for best practices. And then I will also touch upon a few of the LMB flow cytometry projects that are ongoing, where our scientists have been using flow to advance those projects. In 1962, this is actually a drawing by Max Fulwiler, and he's kind of one of the founding fathers of flow cytometry. But in 1962, he joined the Los Alamos Laboratory, New Mexico, with Marvin Vandila. And their primary interest at the time was in monitoring fallout from atmospheric nuclear weapons testing appearing in food products. And after signing the nuclear atmospheric nuclear test ban treaty in 1963, they moved their attention to how they could assist biologists at Los Alamos, and they both became quite interested in the Coulter counter. And basically, the Coulter counter enumerated sized particles suspended in electrolytes, and they were able to then read these sized particles and pulled through a glass tube. And essentially, they were able to count cells. It was a very early instrumentation to count cells. So Fulwiler, in an argument with a pathologist in the group, who, the pathologist was incorrectly interpreting the existence of a population of immature red blood cells. So Fulwiler decided that he was misusing the Coulter counter, and he wanted to prove him wrong. So then he therefore designed a more specific instrumentation that could separate cells based on an electrically generated signal. So Fulwiler had read a paper at the time by Richard Sweet, who developed an ink writing oscillograph, which is now known as the inkjet printer. And he thought this would be a really good idea because the inkjet printer had little small droplets, and he thought that these little droplets would be ideal carriers for a cell. And then he obviously drew up his plans and then patented the device in 1968. So the history of cell sorting actually doesn't stop there. It actually does have some LMB roots, kind of peripherally anyway. So here we have Leonard Herzenberg and of course the great Caesar Milstein. So as I mentioned, Mac and Marvin Vandila were the first kind of founding fathers. But the technique of fluorescent-based cell sorting was actually expanded by Leonard Herzenberg in 1969. So Leonard was a geneticist at Stanford, and he was interested in the addition of a light source in order to sort fluorescently labeled cells. And Fulwiler gave Herzenberg the plans for his cell sorter, where lasers were subsequently added. And upon the invention of monoclonal antibody technology, of course, by Milstein in 1975, this kind of created a perfect marriage between the field of immunology and flow cytometry. So scientists now had the ability to tag antibodies with a fluorescent dye and then identify and fractionate populations of interest for downstream genetic screening or transplantation experiments. And I don't know if any of you know, but Leonard actually did a sabbatical here at the LMB. So yeah, so it does have some kind of peripheral roots at the LMB, which is very neat. There are a broad base of applications that we can use flow sorting for. Everything from, of course, immunology to here molecular biology. It's used routinely in medicine, plant biology, and also marine biology, which is used to characterize the autofluorescent properties of photosynthetic plankton to look at community structure. So I'll talk a little bit about some of the measurable parameters. These are just a few, and obviously it's not an exhaustive. There's many other parameters that can be analyzed. We often look at cell surface and intracellular antigens, protein expression, of course, phosphoproteins, total DNA and RNA content. We also look at transgenic products, GFP, any kind of fluorescent proteins. So in terms of total DNA content, we can look at cell cycle analysis, cell kinetics, proliferation, ploidy or aneuploidy. We also look at apoptosis. We can quantify it. Measurement, we can measure DNA degradation, mitochondrial membrane potential, and caspase activity. And we can also look at things like organelles, where we can mark things like mitochondria with mitochondria tracker dyes. And in kind of more of a translational aspect, we can characterize multi-drug resistance in cancer cells. So there are several components of a flow sorter, the main one being the fluidics. So this is just a schematic about how the fluidics actually work. So it consists of a central core through which the sample fluid is injected, and it's enclosed by an outer sheath, which is actually hydrodynamically focusing the inner stream to focus the cells in a single file. And to be clear here, there's no mixing in ideal conditions anyway. There would be no mixing of the outer core with the inner core of fluid. And of course, it's centered and focused by the Bernoulli effect, and it allows the creation of, like I said, the cells to come down into a single stream and for a single cell interrogation. There are several different types of cell sorters. The two main ones, of course, are the quartz cuvette or cuvette-based sorter versus a jet and air-based sorter. So the sheath fluid that's used, it's typically PBS because that's conductive, and it's of course, a physiological saline, so good for cells. And that is, that comes through, as I mentioned, hydrodynamically focused. But there is a flow chamber above, which is vibrated by means of a piezoelectric transducer. And we can alter the frequency of that transducer anywhere typically between 20 and 90 kilohertz. And this vibration causes the fluid emerging from the nozzle which can be of various sizes to break up into droplets. And then the flow chamber and the flow stream essentially applies a charge to these droplets, and they will pass through a series of two electrostatically charged plates. And those droplets will then be sorted either to the right or left based on their positive or negative or half charges or quarter charges. And so those droplets are then collected into the various collection devices below. We can use a series of different types of tubes, Eppendorf tubes. We can use larger tubes, 50 ml even, and of course into a series of various microtiter plates. So one of the main components of a sorter in terms of its calibration is what's called the drop delay. And this is really essential to make sure that we have a purified population of interest. In other words, as the cell is being interrogated by the laser, the cell of interest that we really want will be correctly chosen and sorted downstream. So since we know the frequency, we can then calculate the time taken for the cell to traverse the distance between the laser interrogation point and the time with which it breaks off to be sorted. And this is known as the drop delay calculation. So this is done every single morning when we set up the sorters, but it's frequently recalibrated throughout the day because, as I will discuss a little bit later, things like temperature changes in the room, any type of small clog or air bubbles can cause a drift in the laser delay and therefore create some serious issues with getting a purified population of cells. So I will talk a little bit now briefly about droplet formation and what it's governed by. So the first is, as I mentioned, the frequency. We can change that frequency from 20 to 90, and of course, as we increase the frequency, the drops, of course, will become smaller, but that also means that we can sort much faster. Also, the amplitude of the transducer, the nozzle diameter, which I will talk about coming up, are obviously the larger the nozzle diameter, the larger the droplet. The sheath pressure, which we can also control very finely, and the phase of the charging pulse. So we do have some factors that affect the drop formation. And the first is the viscosity of the sheath fluid. And as I mentioned, it's temperature dependent. So that's why often in flow core labs, you'll feel quite chilly. We tend to try to keep those temperatures quite low. However, as the temperature changes throughout the day, the viscosity will slightly change. So we have to alter our settings, or not alter the settings, but double check our settings to make sure that we're still in the same spot. Any kind of debris in the nozzle, of course, as you can imagine, cells going through a very small orifice will be perturbed. Those droplets will be perturbed by any debris or junk in the nozzle, which is why we are constantly either sonicating the nozzles to make sure there's nothing in there, or cleaning them. But I'll talk a little bit about how we can mitigate this also with good sample preparation techniques. A complete or partial blockage of the flow cell itself, which of course will also affect any drop formation, and any air bubbles in the fluidic lines or the flow cell. Jet and air sorters tend to be a little bit more sensitive to any air bubbles than the quartz cuvette-based systems, where the interrogation point takes place within the quartz cuvette. In a jet and air system, the interrogation point takes place within mid-air. So those systems tend to be a little bit more sensitive. We have two different types of sorters in the facility. We have both, of course, the quartz cuvette and the jet and air. So we have two Thermo Fisher Bigfoot generaires, and then we have a fusion cell sorter as well, which is a quartz cuvette. We also have an old Synergy sorter, which is still going. It is also a generaire, and that one gets the most cells back of any machine I've ever seen. So if you want a lot of cells back, then you probably want to use the Synergy. Ultimately, a clean and bubble-free sorter is a happy sorter. It's quite a simple concept. So even though it's quite a complicated machine, so making sure that we are constantly running our fluidics and keeping it clean and happy. Some of you are probably interested in the generation of what are the generated drop volumes associated with various different nozzle and sorter types. We can calculate this based on the nozzle diameter, and we actually on our two Thermo Fisher Bigfoot sorters, we can very closely and accurately calculate exactly how much volume will be generated downstream of the sorted particles based on the nozzle size that you're using. So this is, of course, probably quite important for certain downstream experiments where you require very low volume generated, in particular any kind of single cell experiments, RNA sequencing experiments, or 10x experiments where you want the volumes to be very low. So we can calculate this and control this vis-a-vis both the nozzle size, the nozzle diameter, and of course the frequency with which we use. I've talked briefly about nozzle selection, but it's really critical. We use a variety of different nozzle sizes depending on the type of cell or particle that we need to sort. And drop stability and recovery are best when the particle size is less than or equal to the nozzle diameter divided by 5. And it's usually poor if the particle size is greater than the nozzle diameter divided by 4. So for a typical 70 micron nozzle, which we use frequently for things like lymphocytes, we recommend only having cells that are 11 to 14 microns in diameter. And for a 100 micron nozzle, 16 to 20 micron. We also have on our two Thermo Fisher Bigfoot sorters the ability to place in 120 and 150 micron nozzles for much larger cells and potentially more sensitive cells. So as the nozzle size increases, the pressure decreases. So they're sorted at much lower pressures. A typical pressure of a 100 micron nozzle is about 20 psi. And once we get up to about 150 microns, you're looking at about anywhere 3 to 6 psi. So it's quite low. The trade-off to having a larger nozzle and a lower pressure is that you can't sort as quickly. And of course, larger volumes are generated. So there are 3 distinct types of sort modes that are commonly used in flow cytometry. The first is yield, and that's where the number of recovered cells is your primary concern. And this is actually at a cost to, of course, the purity. So each target particle is sorted whether or not a contaminant particle is close enough to be sorted along with the target particle. A yield-based sort typically has a purity of probably about 70%. So it's really, if you're just really kind of looking to enrich a population and you need as many cells as possible, and potentially you want to culture them up and then purify them up later on and do several rounds of this, then this might be the way to go. The most common sort mode that we use is the purity mode. And this is where, of course, purity is the major concern. And you want to make sure that you have purities of over 95%. So the target particles are sorted only if no coincident particle is detected and if no known contaminant particle is included in the droplets. And then finally, the other mode that we use very frequently is a single cell mode, and that's usually used for single cell cloning experiments into plates. And then that essentially means that it is sorting for the maximum achievable purity, but at a great cost to yield. So the presence of any particle, contaminant, or target in a 3-drop envelope surrounding the target particle causes it to be aborted. So this ensures that the cell that is sorted has absolutely no contaminant particle. And in addition to that, there are no empty drops. So this is an example of the 3 different modes. I'll focus a little bit on the purity mode. So if you look at this schematic here, so we have a 3-drop window. The sorted particle needs to fall within the blue lines, so within that area. If there is a non-target particle that falls in any of the area around the red, so close enough that it can drift into the target particle area, it will be aborted. So this is where sample preparation is also really key. If you want to get also high purity, you also want to make sure that your sample preparation is adequate so that it prevents any kind of stickiness in the cells to stick together. The single cell mode is the most stringent because it also requires the cell to be in the center of the drop. So in the purity mode, it can kind of fall anywhere on kind of the side as long as it falls within that blue area of the droplet. It can be in the center, it can be up or down. It will still be sorted. But in the single cell mode, it's absolutely required that it's in the center of the drop. And if there is any kind of contaminant or non-target particle that falls anywhere close to that, it will be aborted. So the takeaway here is that you'll have to think about what you need in terms of downstream experiments. Do you need a lot of cells? What is the actual target population in terms of % expression? What's the nature of that expression? Is it less than 1%? Is it very rare? And do you absolutely require an extreme purity? So these are all questions that you should be asking yourself before you contact us so that we can make sure that we understand your needs and that we will adjust the settings on the sorters to get what you require. So I kind of mentioned these sort envelopes, so 3-drop, 1-drop. What does this mean? So in addition, there are 5 different ways to sort a target. So where the first one is 1 drop is deflected only if the particle is centered in the drop, as I mentioned in the single cell mode. We can also adjust the envelope itself, where a drop is deflected wherever the particle is located in the drop, kind of similar to the purity mode. And then we can go beyond that, where we do 1 or 2 drops deflected, 1 if it's centered, and 2 if it's close enough to the edge that it might drift. Always 2 drops deflected, 1 ahead or behind, and always 3 drops deflected. And to kind of give you a schematic example of what I mean by this when we alter sort envelopes, we do this depending on the nature of the sample. So this is taken from a paper from Van Dyck, where this group adjusted the sort envelope to accommodate filamentous bacteria. And you can see that because of the nature of the bacteria, it's long, it's filamentous, it's not going to fit nicely inside a drop. If you ended up having a 1-drop envelope, you would most certainly at that point eliminate a lot of the sample entirely because it would be too restrictive for the nature shape of that bacteria. So what they did instead was they applied a 3 drops enriched envelope to account for the length and nature of that filamentous bacteria. And as a result, got a far greater bacteria sorted in the end. So again, understanding the nature of your cells in advance. What are their shape? How are they shaped? Are they, are they sticky? Do they tend to contract? Do they tend to kind of have more of a star shape? I mean, I know this sounds not necessarily, very objective, but if you think about kind of the nature in terms of the morphology of the cell, I think it's a lot easier to try to then adjust the settings accordingly on the sorter so that we can get the best possible results. All right, so now a little bit about purity and recovery. So we need to also keep in mind that not all the cells sorted into a tube will be recovered after we spin down the sample post-sort. So we need to consider how many cells of the ones we want are actually deflected into the tube as a measure of sorting efficiency, and that is considered as percent of the sorted or the desired population. And the purity, of course, is a percent of the target population in the sorted cell. So we need to measure how well we've done post-sort. And one way to do that is if we have enough cells at the end of our sort, we rerun the sorted sample to validate the post-sort purity. And I say if possible, because it's not always possible to do this. Of course, if you're doing single cell cloning experiments, that's not possible. If you're sorting maybe only a few hundred cells, that's not necessarily possible. But as a measure of quality control, we always do that just to double check that we are definitely sorting the target population that's needed. In terms of recovery, we always recommend that you spin down your cells post-sort and then decant and then resuspend to be able to get the maximum recovery afterwards. It also depends a little bit on how many cells you're sorting. Of course, if it's only a few hundred or something like this, then it's probably not what you're going to do. But keep in mind, if you're sorting thousands or tens of thousands of cells, always do spin your tubes down before you do any downstream experimentation. And I also recommend that we will provide you with a number on the side of the tube that will indicate approximately how many cells are in there. But it's always good practice to, after spinning down, to actually double check on your own counter, whether it's using a cytometer or some kind of cell counter, just to double check that indeed it should match very closely to the number that we provide to you. So a little bit about verifying the purity and the recovery. So when we actually set up the sorter, we always do a test sort. So we're testing the stability of the side streams before we put on any sample. If the side streams are stable until the sample goes on, it's usually something in the sample that's the problem. And that typically means that the sample itself is too concentrated or the cells are too sticky, they haven't been filtered. So when the recovery is less than expected, it's very likely that the sample did not follow up with sound distribution through the sorter. So as I mentioned, the cells are too sticky, so they stuck together, which caused some aborted events. There's too much serum in your sample, so too much serum or too high amounts of protein, such as FCS, anything over, let's say 5%, can cause stickiness. Sometimes the cells are not very happy when they're brought to us. So in that instance, you may want to use or add a little bit of DNase to your sample media, just to make sure that any DNA that's released is degraded and doesn't cause any stickiness in your sample. Another reason for issues could be that an inappropriate nozzle selection has resulted in an unstable droplet generation. So this is where it's really important that typically we have quite a bit of information already in terms of the particle size. But if we're working with something new, it's really important that you let us know what approximately the size of the cell is. Because if you imagine we're trying to squeeze through, a 40 micron cell through a 70 micron nozzle, it's not going to work very well. So if you're not sure, you can always use some imaging techniques or microscopy, or to verify your cell particle against a bead standard, which is very common, particularly if your cells are much smaller. It's also very important to verify anything that's very small on the other extreme end of things, because the smaller a particle is, the more likely you will get something called a swarming effect at the laser intercept point if you're sorting things too quickly. So in other words, you will get potentially more than one particle or a swarm of particles that will hit the laser simultaneously. And therefore that will reduce our ability to be able to isolate your populations of interest. As we all understand that this is a single cell, a high throughput single cell technology. So we really need to make sure that we're adjusting our settings so that the cells are really only coming in one at a time. All right, so a little bit about sample preparation now. The buffer choice is really, really key. I can't emphasize this enough. It really requires some attention to detail in advance. And when you're fractionating out cells of interest, you want to actually do this in a timely way to preserve viability. And of course, any kind of downstream functionality, particularly if you're planning on expanding anything in culture for downstream assays, for example, like in cytokine production. So it is really important to have a really excellent single cell suspension to ensure the best possible flow while minimizing doublets. So that's more than one cell suck in the interrogation point. So we typically recommend DPBS or HBSS, calcium magnesium free for most preparations, potentially not for neurons, for example, with about 10 to 25 millimolar HEPs and 1 to 2% serum maximum. What we don't recommend are things like RPMI or DMEM. And one of the reasons why we don't recommend that is that those buffers are a different buffer than the sheath fluid that is being hydrodynamically focused on the outside. And by design, they require a 5% CO2 to maintain physiological pH. So if you're sorting for long periods, that's not potentially going to result in a very high viability. And the other thing that you want to think about is also adding 1 millimolar EDTA in addition to this, the HEPs, because that sometimes 1 to 2 millimolars max for very sticky cells that are adherent, for example, so that it makes sure that they stay in a nice single cell for interrogation. And the other thing to think about is to protect the sample from any excessive light or any kind of exposure to light. Because that will also, some of these preparations or media sample preparations can be light sensitive. All right, so a bit of advice for preparation guidelines between adherent cells and cells isolated from solid tissues. So often any kind of cells that are isolated from solid tissues require some kind of more, let's say, mechanical and enzymatic digestion. The first is to make sure you inactivate your trypsin or with soybean trypsin inhibitor, or use accutase, obviously to maintain viability. Any cells grown in media with serum should be rinsed twice with PBS prior to dissociation. You should always test your dissociation techniques for downstream viability. We can help you with that if you haven't already been trained on any of the analyzers in the facility. We can, of course, just double check the viability if you're kind of testing any dissociation techniques upstream from a very important cell sort. So you don't want to put all your eggs in one basket. You should probably do any kind of pre-optimization experiments first if you're unsure. You definitely want to test any non-enzymatic buffers relying on any chelating agents as well. And then the highest quality reagents, of course, should be used. And just keep in mind that if you're using collagenase, it's dependent on calcium for activation. All right, a little bit about the collection tubes that we're using or that we recommend and some of the buffers that we recommend. So the first is that it's the best practice to pre-coat collection tubes with serum. And one of the reasons why we recommend this is, first, when we're calibrating the sorters, particularly that we're calibrating the location of where the side streams that are generated, where they are hitting inside the collection buffers. We aim to have those side streams hit the exact center of the collection tube. However, if there's any kind of perturbation throughout the sort, it might kind of slightly angle off. And if it hits the side of the tube, then it's hitting hard plastic, and it will, in terms of the impact, but also in terms of just drying out. So the droplets generated are very small in terms of volume. So if you're coating and pre-coat the collection tubes with serum, then it gives a chance for that cell to kind of slide down into the center of the collection tube first, so that it maintains viability. Some people actually do this and pre-coat the collection tubes overnight by rotating them overnight. The second tip is to use the same sample buffer for the collection tubes with a little bit higher percentage of serum than you would in your sample prep. So this also ensures that once the cell goes through the sorter, if you think about it, it's fairly violent. It's a bit shocking that we still are able to get viable cells at the end, but you're really increasing the chances that your cell will maintain its viability and it's landing in a nice, happy kind of, serum environment. So, but the other reason why we always recommend the same sample buffer is that if you have two different sample buffers, if you have the collection buffer that is a different type of buffer from what your cells are in, then it's going to essentially, there would be a mismatch potentially in pH. So for example, any kind of bicarbonate media, if you're using a bicarbonate media to the collection tubes, as the sort buffer is added, it's going to increase your pH and it's going to cause undesirable salts to form and, of course, result in some reduced viability. Media added to any wells should be heat buffered and conditioned beforehand. Again, don't use unbuffered bicarbonate media. And for very rare sorts, we recommend into either wells into plates or 1.5 or even micro PCR tubes for very rare subsets. Ultimately, this will ensure higher recovery. If you imagine a very rare population kind of having to traverse into a very large 15 ml tube and trying to recover those cells from a 15 ml tube versus a very small Eppendorf, I think you can appreciate that using a smaller collection device will help your recovery. And finally, we recommend polypropylene fax tubes over polystyrene for the collection because the cells will adhere less to the walls of the tube. The other thing with polystyrene tubes is that they can result in a buildup of charge in the deflected drops on the surface. And sometimes as the cells are being sorted and as the media in the collected tubes is increasing in volume. That can cause some charge to build up, and then you get what's called kind of a jumping drop effect, where you have repulsion. So for example, you have the cells on the right that are positively charged, and then another population on the left that's negatively charged. You're going to get some jumping drops there, and then causing some contamination between your collection tubes. We mitigate that a little bit on the Bigfoot sorters by applying what's called a polarity flipping. So essentially on the Bigfoot sorters, we can swap the charge throughout the sort so that there is the net charge that is in the collection device is neutral. And we also mitigate that by ensuring that the collection tubes don't get overfilled. So we always leave quite a bit of a buffer. So if we have a 5 ml fax tube, we wouldn't sort all the way up to 4 ml of volume. We'd probably prefer to sort more like 3.5, just to mitigate any kind of increase or decrease of purity at the end. All right, so now I will talk about 3 different projects that have been going on or are ongoing actually in the LMB. And I will highlight 3 different projects from 3 different divisions. The first is one by Miranda Robbins in the Zlatich lab, and she is looking at molecular changes required for memory formation in Drosophila melanogaster. The really challenging part of this project is that she needs to, of course, remove these neurons and image them first, and before she brings them for sorting. So her project involves determining whether different dopaminergic neurons and their postsynaptic cells involved in reward learning have different naive transcriptomes, so before learning. And she wants to help understand how this complexity of molecular signaling can occur in the circuit. For example, how many transmitters or receptors, and some sensitivity to calcium. So as I mentioned, it's quite challenging, first and foremost from a sample preparation upstream to bringing them for sorting. But the other extremely challenging part of this project is actually getting enough cells. So she's labeled these cells with either TD tomato or neon green. And you can appreciate here, I think, from the plot that they're quite rare. So again, making sure that we are optimizing the settings on the sorter to try to get her as many purified cells of interest downstream for her experiments are really key. The second project that's ongoing in the LMB and with the Flow Cytometry Facility is one by Chris Kuo in the Baum lab. And of course, Chris is looking, and the Baum lab in general is looking at cell division in archaea. And these are thermoacidophilic archaea and a facultative autotroph found in thermal soils and hot springs. And the goal of one of Chris's projects is to isolate these archaea at different substages during cell division, and to pinpoint at which substage the transcription of cell division genes are turned off. And subsequent qPCR analysis on the sorted cells showed that the transcript abundance of the division genes peak when the division ring is constricting and rapidly turned off upon completion of division. So this project is also challenging, but from a completely different perspective, mostly because archaea are very small. So we need to make sure that we're able to identify them within the resolution limits of the sorter. But also, we need to ensure that our alignment of the machine is very, very tight. The alignment of the lasers to ensure that we're getting a very tight cell cycle peaks, and that we're able to subsequently sort the different populations throughout the cell cycle in a very purified manner. So, and that's been ongoing for some time in the facility. And then the final project that I wanted to just highlight briefly is one from Michelle Coe in the McKenzie lab. And her project seeks to understand how the enteric nervous system and immune cells interact to regulate defense responses against helminths. And in the intestine, there's thousands of neurons and glia that coordinate all the necessary functions of digestion and nutrient absorption and defense. And she's specifically focused on a population of enteric neurons that express molecules known to regulate lymphoid type 2 cells, ILC2 cells. And it is very difficult to study this population of neurons in a heterogeneous mix of neurons and glia, and this is where cell sorting comes in. So we purify these neurons of interest using a transgenic mouse strain where these neurons express TDRFP, and then she cultures them downstream to perform transcriptomics and for in vitro assays. And this is a lovely image provided by Michelle of the neurons day 19 post-sort. So you can see that they've definitely are nice and happy and alive and are growing well. So neurons are not necessarily the easiest cells to sort, especially once they've been dissociated into single cell suspension. And we've been very happy to be able to work closely with Michelle and the McKenzie lab to be able to optimize this experiment. This is also an example of where we have used the polarity flipping option on the Bigfoot sorters so that the neurons, they are charged as they're sorting. We can never avoid that. But as the cells accumulate in the collection tube, the net charge is neutral, so they're not always being activated. All right, I will end with some future developments briefly in specifically related to cell sorting. And the first is image-enabled cell sorting, where we couple microscopy to cell sorting techniques. And one of the things that came out recently, and you can go and read this paper in Science that was published in 2022, where in collaboration with BD Biosciences, this group developed an imaging cell sorter where they use fluorescence imaging using radio frequency tagged emission. It's a traditional cuvette-based droplet sorter, and it has very low latency signal processing and sorting electronics. So the really amazing thing about the development of this machine is that it produces an image of your cells, but it's still able to maintain a very high speed of sorting. So up to, they claim, 15,000 events per second. So most of the imaging sorters that were kind of in development, or several groups that were trying to develop this, have always had problems with, of course, the speed of sorting, so that they could get a blur-free image. So this is just taken from the paper. This is a schematic of how the system works. And so to enable the blur-free imaging at a flow speed fairly high of 1.1 meters per second, it uses this fire approach, which produces an array of 104 laser spots across a 60 micron beam within the core stream of the sorter cuvette. And each of those spots is modulated at a unique radio frequency. And so that's able to then create an image of the cells. As you can see here, they actually did a CRISPR screen and they marked various organelles here, as you can see in green. And so, yeah, this is quite the development. If you can imagine now that we're coupling spatial and subcellular localization with cell sorting, it really opens up a whole new area of potential applications. because we can now, not only are we just looking at mean fluorescence intensity, but we're able to actually localize where that intensity and where that fluorescence is coming from. So it's really quite an amazing development. And I anticipate, not just me, but the community anticipates that this will only move forward into greater resolution and abilities to fractionate out cells with more accuracy. A couple of the applications. The first is using cell painting or morphological profiling. We can use this for high throughput functional genomic screening, as I mentioned, CRISPR screening. Things like quantitative analysis of internalized bacteria and protozoan parasites, more analysis of asymmetric cell division, and finally, even some analysis of cell interactions and immune synapse. There's a few limitations of the image-enabled cell sorting at the moment. The first is that the image part is only consists of one laser, so you can only use fluorophores that are excited off the 488 nanometer laser. And it has 4 fluorescent detectors, but you're not able to compensate those out. So essentially it means that you're only able to use one color for the imaging part, potentially 2 if you use something that's more in the far red, and you don't have to spectrally compensate those 2, but that's kind of a major limitation at the moment. In terms of the resolution that you're getting, it's a 10 times magnification with a 0.3 NA objective lens, which realistically results in an approximately 3 micron resolution. So the image itself is quite blurry, but it's still moving forward. So this is good. It's a good development, but you need to be realistic about what you're able to actually resolve. Certain applications will require cell fixation, which is obviously not ideal if you want to fractionate out things for downstream experiments that require some functionality and viability. And as we move forward with these kinds of technologies, it's going to really require more in-depth knowledge of both imaging and cytometry modalities. So I anticipate that flow cytometry, the flow cytometry and imaging community will have to kind of work more closely together to ensure that all of the technical experts going forward will be trained appropriately in both techniques. The next frontier is really high resolution multispectral cytometric cell sorting. We have currently a collaboration with James Manton in the Flow facility, and our postdoc, Bill Flanagan, is currently working on a system where he will expand Akash Kumar's development of a light sheet multispectral imaging microscope to couple that to a cell sorter. So we're hoping to increase the resolution that's currently available on the market, but also most importantly, we are hoping to include the ability to be able to do more than one fluorophore. And that will really be key to kind of push this technological development forward, where we're able to potentially mark more than just one organelle, but maybe up to 4 or 5 simultaneously, and get better resolution as well. So yeah, it's really exciting times, I think, in the community. All right, finally, a little bit about our team and what we're here for and how we can help you. So I think it goes without saying that we're really here to help you make the most of your experiments. We're very serious about what we do, and we really want to make sure that we get the best results for you, and we're generating the absolute highest quality data. And we actually welcome working with challenging samples. It makes it really fun. I love getting all of this challenging stuff in the LMB. It's what makes being here so amazing, actually. Both Fan and I have a depth and breadth of expertise across various technologies and applications. And if your experiments are part of a larger pipeline across facilities, so for example, if you are doing any downstream, if you're doing sorting for any downstream purification from mass spec, or any kind of imaging, for example, cryo-EM, please consider the power of cross-facility communication to facilitate and accelerate your success. I currently have also a collaboration ongoing with Kat Franco in mass spec and Aidan Zetto to look at some more details about whether or not certain polymer dyes can cause some issues with a downstream mass spec. So this is a good example where cross-facility communication can really help to troubleshoot potentially anything that you're having issues with. And if you make use of the technology and expertise in the facility, please make sure you remember to formally acknowledge the facility in any publications. I think this is incredibly important. We need to report back to our funders, the MRC, to make sure that they understand that money that they give us is going being put to good use. And so we use these publications to show that. So please do acknowledge us in any publications. And that's it. Any questions? Thank you very much.